Interactomes. This is the non-covalent interactions that function within a cell. Nobody knows how a viable cell emerges from massive combinatorial complexity of its molecular components. And of course, nobody's ever synthetically mimicked it. An interactome is the whole set of molecular interactions in a particular cell. If one merely considers protein-protein interaction combinations in just a single yeast cell, the result is an estimated 10 to the 79 billion combinations. That's estimated by these folks at Johns Hopkins and Brussels, all right? That's not my estimation. These are biophysicists, all right? So just to give you an idea of how big that number is, that's 10 to the 90th is the number of elemental particles in the universe. That's 10 to the 90th. This is 10 to the 79 billion. That's a big, big number. This was the first generation motor that we put in the car. It worked, but it only rotated 1.8 revolutions per hour. When we rebuild it without that sulfur atom there, then that 5-member ring, then it goes 3 million rotations per second. All right, so small changes can make a big difference. Well, what do you do? Well, what you do is you just come here and you erase that sulfur and you do that. Now that you can do on a blackboard. But this took us back to step 1. This is what I'm talking about. When something you're going along and you want to build and you say, oh, I'm just near life. If you have one thing wrong, it doesn't work. So, well, you just modify this and it'll work. All right, go back to the beginning. Go back 1 billion years to the beginning. It's too hard. Nobody knows how this thing is solved. You can't build the molecules. Next slide. And then, so remember, these just to make the molecules, the simple molecules. You got to have the carbohydrates, you have to have the lipids, you have to have the nucleic acids, you have to have the proteins. Even if you make an amino acid, how do you hook the amino acids together? Amino acids don't hook together by themselves. We're not even talking about order. How do you just get them to hook? I mean, somebody sent me a paper just last night. I mean, here's a paper just came out on self-folding molecules. Well, duh, that doesn't prove anything. You build a molecule, it folds up. Big deal. How do you get these things to hook together in the first place? Nobody knows. That's with amino acids. How about with nucleotides? You want to get these things to hook up. People will make oh, you see, we got a nucleotide. We got DNA. No, you don't have DNA. You got to get these nucleotides to hook up. Look at a DNA synthesizer. How many steps of blocking, protection, deprotection you need to do the complex chemistry just to get one linkage made? Nobody knows how. You can throw all these together in a flash. People say, well, the concentration of nucleotides was very high. I've read this in high school books. It was very high. And these things came together. You calculate the concentration, the world would have to be raining nucleotides. And even if it were raining nucleotides at those concentrations, they wouldn't hook up. They don't hook up. You need enzymes to hook them up. But this is prebiotic. There's no enzymes here. Or you need very complex DNA synthesizers. Those weren't there either on early Earth. Now you have to assemble it. So say we had the molecules, how can you get them to assemble? So here's the assembly experiments. A protocell is a self-organized, endogenously ordered spherical collection of lipids proposed as a stepping stone to origin of life. So that's what a protocell is. But most so-called protocell assembly experiments in origin of life research can be summed up by a protocol analogous to this. You purchase homochiral diacetyl lipids from a chemical company, or you synthesize, scramble lipids from smaller molecules. Add those lipids to water and observe a small amount of it to form simple and expected thermodynamically driven assembly of those lipids into synthetic bilayer vesicles upon agitation. Sometimes the researchers will add other molecules that get engulfed by those vesicles. Then you publish a paper claiming that the synthetic vesicle is a protocell and suggestive of early forms of life. Engage with the media, hype it up, watch the layperson be misled. Every assembly experiment, I'm telling you, can be summarized in this. So the complex cellular membrane. So this is actually what the membrane of a cell looks like when it's made really, really simple in cartoon form. So researchers have identified thousands, actually 40,000 different lipids now have been identified in cell membranes. Not just one. Every protocell experiment, they use one type of lipid. Actually, a cell is made up of thousands of different types. Mixtures of monoacyl lipids destabilize the system. So when they have their monoacyl lipids, those will destabilize the systems. You can't use those. Lipid bilayers surround subcellular organelles such as nuclei and mitochondria, which are themselves microsystem assemblies. Each of these has their own lipid composition. So this is a lipid bilayer. You have some pointing out toward water, some pointing in toward the water on the interior of the cell. And the outside ones are different than the inside ones. Nobody knows how that was done. Nobody knows. Clueless. Nobody knows how that was done. Every protocell experiment just uses homogeneity throughout the whole thing. And so it's not really a protocell. It's not really reminiscent of a cell. Now, within each one of those, you have other organelles like nuclei and mitochondria that have their own bilayer assemblies with their own constitution. Different than what's on the outside of the cell. Nobody knows how that was done. Plus, these have these proteins. So there's this non-symmetric distribution. Then there's proteins that go through here. These are ionophores. That allows certain things into the cell and certain things out. Without that, you can't let anything into the cell. It's going to kill the cell. It has very discrete sensors that allow certain molecules to get in, certain other molecules to get out. It has ionophores that allow certain ions in, certain ions out to keep the ionic concentration at a certain level for maintenance of that cell. As soon as that ionic concentration gets off, you know what happens? Boom! The cell explodes because the ionic concentration has gotten off. It blebs and explodes. Nobody knows how that is done. None of the protocell experiments, none of them have these proteins going through them as these control gateways. Then there's also lipid bilayers have a vast number of carbohydrate appendages. So off of these, this is a carbohydrate appendage. It's called a glycan. The artist is just showing us a few of these. A cell is covered with these. This is how cells recognize each other. How does one cell recognize another cell? By these carbohydrate assemblies that they recognize each other. They have this recognition patterns. And so you can tell one cell from another. And so if you just take nucleotides, if you just take DNA is so complex and it has so much information. Look, if you have 6 A bases, A, how can you arrange those? What are the different ways you can arrange it? Just that one, A. That's it. One way you can arrange 6 bases. Look at carbohydrates. If you just take the carbohydrate D-pyranose, just a standard carbohydrate, just D-pyranose, you have 6 of those. Has over 1 trillion ways it can assemble. 1 trillion ways it can assemble just with 6 of them. These have much more than 6. And if it's not assembled right, guess what? The cell dies. You change any one of the carbohydrates, it results in cell death. There is much more information stored in carbohydrate assembly than in DNA. Yeah. Much more information can be stored in carbohydrates. You want to build a massive computer? Build it based on carbohydrate assembly. Much better than DNA assembly. It's just a carbohydrate assembly is really hard to control. Nobody knows how to control this. Nobody. But somehow, on a prebiotic Earth, with nobody around, under a rock, it figured this out. How do origin of life researchers address this problem? They don't. They don't. Next slide. Interactomes. This is the non-covalent interactions that function within a cell. Nobody knows how a viable cell emerges from massive combinatorial complexity of its molecular components. And of course, nobody's ever synthetically mimicked it. An interactome is the whole set of molecular interactions in a particular cell. If one merely considers protein-protein interaction combinations in just a single yeast cell, the result is an estimated 10 to the 79 billion combinations. That's estimated by these folks at Johns Hopkins and Brussels, all right? That's not my estimation. These are biophysicists, all right? So just to give you an idea of how big that number is, that's 10 to the 90th is the number of elemental particles in the universe. That's 10 to the 90th. This is 10 to the 79 billion. That's a big, big number.